I've been working on this genetic disease called familial hypercholesterolemia. That's a bit of a mouthful, so I'll call it FH for short. So people have inherited a defect, which means they can't clear cholesterol, LDL cholesterol, from the blood. The cholesterol builds up in their blood. It causes them to have heart disease at a very early age. And because it's genetic, it runs in families, which means once we've found an FH patient, we can test their relatives very easily, their brothers and sisters and their children, and roughly half of them will also have FH. So once we've found them, we can then give them statins, we can advise them to stop smoking and things like this.
So I'm a urologist. I'm unusual in that I specialise just in prostate and mainly these days in prostate cancer. We're interested in improving the diagnostic pathway for men with prostate cancer, which has been troublesome to date, largely based on the unreliability of the tests that we've used, which have been PSA, prostate-specific antigen, and also the biopsy of the prostate, which is one that's done through the rectum and is blind to the location of the tumour.
I think the most exciting thing that has recently happened is that we have developed a new method which allows us to recover pathogens from clinical samples without the need for culture or even to amplify them in any way, and we can then sequence these pathogens and obtain whole pathogen genome sequences, and that gives us very good information about how related pathogens are, and we can then determine whether people are infecting each other, how the pathogen is spreading, how the pathogen is evolving, whether the pathogen has developed characteristics that then lead to a more severe infection, and so it's opening a huge area of research that really wasn't properly available, especially for pathogens that couldn't be cultured and couldn't be amplified prior to sequencing.
My team is particularly interested in identifying new ways of early detection of breast and gynecological cancers, in particular ovarian cancer. We have identified changes in the genetic information of cancer cells called the DNA, which is able to identify, or which we aim to identify women at the earliest possible stage by developing a blood test. These DNA changes are called DNA methylation or epigenetic alterations, which are unique to cancer cells and are not present in normal cells. The tumor cells shed their DNA once the tumor cell has died into the bloodstream, and we aim to develop a test which picks up these different epigenetic changes in the bloodstream well in advance of a clinical diagnosis of patients.
Carrying out this sort of work really bridges the gap between the patients who are seen in the lipid clinic and the molecular biology, what's going on, if you like, really at the heart of the cell, at the heart of the individual. So by knowing the molecular causes in an individual, why they've got high cholesterol, we can actually give them much better information. The other thing is, if we can find a completely new gene where a mutation is causing FH, that might then give us a clue about how to develop a new drug. We would then want to turn this new gene on or off, and that really would be very exciting if we could find a new gene causing FH.
The diagnostic pathway results in cancers being missed, in very, very small non-lethal cancers being identified, and therefore labelling a patient with cancer from which they probably won't benefit. This is called overdiagnosis, and that results in overtreatment. In other words, patients having treatment that they won't benefit from. This is a difficult concept, but is prevalent in many of the common cancers, such as breast cancer and particularly the prostate. So what we're after is developing a test that identifies just the lethal cancers and overlooks the cancers that most of us will have as we age that actually won't cause a problem to us during our lifetime.
There are many things we want to do. We want to do much more surveying of how pathogens transmit. We've started with viruses, which is the field from which I come. We're moving on to look at non-culturable bacteria. such as chlamydia and mycoplasma genitalium. We're starting to look at those. We're also starting to ask whether we can actually recover some organisms quicker rather than waiting for them to grow, for example Mycobacterium tuberculosis. And all of those, if we're able to sequence pathogen genomes directly from clinical material, we'll be able to actually ask much more finely tuned questions about how things transmit, who infects whom, why some people don't become infected and why others do, and susceptibility and that sort of thing.
So our new diagnostic strategy, which is largely based on imaging, on MRI, would result in fewer cancers being identified, but the cancers that we do identify are the cancers that are most likely to cause problems and perhaps result in an early death. Also, we can reduce the number of men that have biopsies, and we think that possibly with time, if the research fulfills our expectation, about 500,000 men a year in Europe alone may be able to avoid an unnecessary biopsy.
The relationship between the hospital and the university is critically important for delivering the type of research that I'm currently undertaking. UCLH has a major focus on cancer. Cancer is one of the 4 BRC themes, and the hospital is invested heavily in the infrastructure for the treatment of cancer.
UCL and UCLH is obviously a very exciting place where to work, simply because it's a constant collaboration with amazing scientists, clinicians, but also, and maybe most importantly, the patients, because we shouldn't forget that none of this research would be possible without the patients and without the patients' samples, such as skin biopsy, for instance. We work a lot with patient fibroblasts in my lab. So this is very valuable.
We're also opposite a multidisciplinary university which has excellent mathematics, biochemistry, chemistry. So we have a lot of relationships with the university. So I think in total the infrastructure for performing experimental cancer medicine is outstanding, and I think is an ideal environment for researchers such as myself to pursue their work.
To work at University College London and the University College London Hospital has a lot of advantages compared to other centres. We have managed to centralize services for both breast and gynecological cancers, now in particular within the University College London Partner Initiative and within London Cancer. The volume of patients and the close proximity to the laboratories makes London, and in particular UCL, an extremely good place to conduct and lead this research.
It's provided us with clinical colleagues. We've sort of got to know people that through the BRC who've helped with our research. I've had funding personally from the BRC, which has given me time to spend on this research. And I've had a postdoc who's an evolutionary biologist funded on a project, a small project grant from the BRC, and that's allowed us to kickstart these sort of projects that I've been talking about.
Well, we've just recently been given support from the BRC to fund someone working in the laboratory who will be sequencing genes and to be doing finding the mutations in all these FH patients, and then a nurse who will go out and find the patients and recruit them in for all these different studies. So without the BRC support, we wouldn't be able to really take these results forward and to see how much further we can go.